This is a production of Cornell University.
Thank you very much, Alina. The one thing, I actually spent a year working for the USDA too. So a finger in many pots. Okay, so this first slide is to characterize basically my good fortune. I was coming back from when the Arabidopsis meeting was in Beijing, and I had like 48 hours in Paris. And so I went to the Louvre, and they had this thing where they were taking modern art and, oh, let's see, yeah, I got that. And, well, I don't know, this thing, what have I done here? How can I have broken it already? So I've broken this already. But anyway, they juxtaposing modern art with all the ancient stuff. And we were actually on one of those moving runways out of there. Yeah. And we saw, you know, all these life-size plaster of Paris people wearing lab coats and holding clocks, and I thought this is just so perfect for my lab. So we actually had to go out and come back in so I could walk in there and take a picture of all these people. So that is not a group meeting, that's art. Let's see. Okay, so the reason for my fascination with clocks and the whole premise of the field is that a clock that matches the environmental oscillation will enhance fitness. Because, of course, the rotation of the Earth on its axis means that life has evolved in a world where the environment changes drastically, day and night, but completely predictably. And so if the clock is an entity that allows the organism to coordinate its biology with this daily oscillation in the environment around it. So that's what we're working on. And nowhere is this better illustrated than in this movie that some of you may have seen from Roger Hangarter make this. This is a sunflower plant growing in Roger's backyard. And this is the day in the life of this sunflower. And so this is east and this is west. And it's Indiana, so not much is going to happen. But over time, the shadows will get longer, and then it'll be illuminated at night. The action is all at night, and it'll be illuminated by a single bulb over his back porch. But you can see the solar tracking. It's been tracking and keeping its leaves oriented. And now, immediately, it completely reconfigures. And there's sort of two amazing things here. One is it knows where the sun's going to come up and where breakfast's going to be. And it knows when it's going to happen, and it's totally ready. So this is the best example of dawn anticipation that I have ever seen. And this is really what clocks are about, to enable you to anticipate what's coming next and adjust your biology to be appropriate to what's happening next. Now, I always like to put this in particularly when it's not a plant audience, because I like to sort of poke the animal folks in the eye a bit. But given that it's largely a plant audience, this hopefully will make you all feel better about yourselves. Because plants feature almost entirely the history of study of circadian rhythms is the study of plant circadian rhythms. In the Western canon, at least, the first writings on clocks are those of Androsthenes, who was a scribe who went off with Alexander the Great to define and conquer the known world. And he observed leaf movement of a tree, almost certainly a tamarind, on an island in the Red Sea, now we call it Bahrain. There was no indication that they had any sense that this was an endogenous, self-sustaining leaf movement, but this was the first writing. Science moved a little slower back then. Some 2 millennia later, a French astronomer finally did the experiment to ask, is this endogenous? And he took a sensitive heliotrope, almost certainly a mimosa, and just put it in the dark and asked, do the sleep movements of the leaves persist? And they did. He was quite prescient because he suggested there might be a relationship of this behavior in plants to the observed sleep behaviors in bedridden human patients in hospitals. And of course, he was quite right, both product of a clock. Again, science moved slowly. 30 years later, 2 other authors published books repeating those observations, getting whole books out here, but they excluded temperature as having driven these rhythms. And then finally, 80 years later, de Candolle finally got quantitative. And up to this point, people had said, okay, leaves are moving or whatever. They have a circadian period, a daily period. But finally, de Candolle quantified the period and determined that the free-running period was not precisely 24 hours. In mimosa, it's about 22 to 23 hours. And when I entered the field, we were still at the end of a long debate with whether or not there was such a thing as the circadian clock, or rather, were these overt phenotypes that people observed the result of some response to some subtle and undetected geophysical cue. And of course, the answer came in here because the period wasn't 24 hours, so it couldn't have been a subtle geophysical cue, because that would have had a 24-hour period like the rotation of the Earth on its axis. So it had to be endogenous. People still didn't believe until they finally put Neurospora up in Skylab, removed it from the Earth cycle, and it continued to show its daily rhythm and cannadiation. So, but up until now, everything's all in plants, and it wasn't until, I think it was 1898, when finally they observed rhythms and pigmentation in some crabs. And they didn't observe rhythmic behavior in rodents, rats and mice, until about 1924 or something like that. So basically, the history of clocks is the history of plant clocks, and animals are recent interlopers. In plants, basically, whatever you're doing, there's a good chance that there will be an influence of time of day on that phenomenon, which is really an inconvenient truth, but it's my assertion to you. And so we know that growth responses, elongation growth happens, it's gated to particular times of day. There are many examples of metabolic gating. Of course, we know the clock influences photoperiodic flowering. And recent events, and I'll show you a couple of examples, have provided evidence that the ability to perceive and mount a response to biotic or abiotic stresses can be a function of the time of day at which that stress is encountered. So they're really quite pervasive. Do rhythms matter? I want to give you two examples where they do matter. The first, I think, is in many ways the coolest. This is work from Alexander Graf, who was a student with Alison Smith. And it has to do with starch metabolism. Basically, a Arabidopsis stores its excess photosynthate in the day as starch, and then at night degrades that to provide carbon for growth and respiration. And of course, it would be bad if you burned it all up and ran out. What happens is that at dusk, the plant makes a calculation, how much starch did I accumulate today, consults its clock, and anticipates when dawn is going to be, and adjusts its starch degradation rates to be linear, such that starch reserves are just about but not quite exhausted at dawn. If you fool it by delaying dawn, the plant does this linear burn, runs out of starch, induces a carbon starvation response, and growth falls off dramatically. So here's one great example that may be why it's important to have a clock to do this calculation. And of course, the title of one of the reviews was Plants Do Arithmetic Division. I just love that. The other one, here's another one for those of you who like caterpillars. So cabbage loopers like to devour Arabidopsis. And so Goodspeed et al. In Janet Bram's lab did some totally cool experiment. They entrained the loopers and the plants in phase with one another. So they both think day is when day is. And there's an activity rhythm and a feeding rhythm. The Trichoplusianae are active during the daytime. And if they're in phase with the plants, everybody thinks it's daytime. And there's some herbivory and some growth of the loopers, but the plants survive. If, however, you entrain the plants and the loopers out of phase with one another, such that the looper is active when it thinks it's day, but the plant thinks it's night, the plants are devastated, and the loopers proliferate and get big and fat. And of course, now what we know is that they're cycling jasmonic acid levels, and the plant, of course, there's a trade-off between defense and growth, and the plant is just making JA when it anticipates the activity rhythm of the looper. And it seems if you disrupt that relationship, it's bad news for the plants. So here are two examples, but there are many, many other examples. I mean, we don't know exactly, there's no one thing you can look at and say, this is why clocks enhance fitness. So far, it's probably an incremental improvement on lots of different processes. And it, which you think is most important, what do we call that? An ascertainment bias. It's whatever you think is important, and you look at it and you'll find it, maybe. So to define some terms, so circadian, the term means, it's from the Latin, approximately, circa, approximately, dies day. So these are rhythms that have a period length of approximately, but not exactly 24 hours. And they're endogenous. They persist in the absence of environmental time cues. But of course, we live in a world that's replete with environmental time cues. And so clocks are entrainable to environmental time cues, chiefly light-dark cycles, but also temperature cycles. In the case of you folks, most of whom I think are humans, if you fly to Japan, you're going to be jet-lagged. The light-dark cycles, take a walk at noon, that'll help you entrain. But also social cues, taking your meals on time, having a beer at the end of the day, all these sorts of social cues will help to entrain your clock. So let's see. A couple of terms that I'll mention a bit. I won't mention amplitude much. It's your sine wave physics from high school. Half the peak to trough distance is one amplitude. In entraining conditions, when we're providing a light-dark cycle, the period will be entrained to 24 hours. If you're released into continuous conditions, the gray is when the lights would have gone off had they gone off. You'll see revealed the free running period. In this example, it's a little longer than 24, about 25 hours. So traditionally, we thought about the clock in sort of a linear cascade. There's a clock. It's entrained through input pathways, light, temperature, imbibition. In this case, I mean the hydration of the seed. But that's the social cues of taking your beer on time will help you entrain. And then there are output rhythms. Gene expression I'll talk about. I'll talk a lot about leaf movements, somatal aperture, other things. But this linear pathway is not sufficient to think about this because we know there's gating. That is, the plant, through clock-regulated expression, for example, of phytochromes, modulates its own sensitivity to light. So, and we can think about that with a plant, but also with us. A pulse of light applied to you at noon doesn't affect your clock. Of course, there's light at noon. But that same pulse of light applied to you in the pre-dawn hours, you think, oh my goodness, I've overslept. The lights are on already. And you advance your clock to match what you've now perceived as a new dawn. That same pulse of light applied to you after dusk, you think, oh, I thought the sun went down, but it just went down again, and you delay your clock. So it's very contextual. It's time of day dependent, and that's this gating phenomenon. In addition, many of the output pathways have intermediates, and it's now becoming clear in a number of ways that various intermediates in these output pathways, metabolites like sugars, nutrients like organic nitrogen, iron, copper, are actually feeding back, again, to modulate clock function. And so if you're setting up all these to be appropriate at time of day and they're interacting with the rest of the organism, of course you're monitoring key metabolites to try and fine-tune the clock. So it's not a linear pathway at all. It's a network pathway. And you have to think of the clock, I think, as an integrator of cellular metabolism and organismal physiology. At least that's the way I'm trying to think about it. Work from a lot of labs, among them that of Steve Kay, in Arabidopsis have gone a long way towards defining the architecture and the components and the interrelationships of these components in the plant clock. And so like all these complicated machines, it gets complex and then people simplify it, then it gets complex again. We're in a period of slight simplification, but basically there are a whole bunch of transcription factors, most of which are repressors. At dawn, CCA1 and LHY, two single mid-domain transcription factors, are abundant, and they repress a whole bunch of other things. So there's this morning component. There are also evening components, and these are arrayed according to times of day. So there's an evening complex and TOC1, and these guys are also repressors. What happens is, after a period of time, certain transcriptional activators turn on this bridging group, and they turn off this, and so on. And you with all this interrelated on-off, you can build up a stable 24-hour oscillation with these feedback loops. And depending on your love of complexity, you can add way more arrows and more components to this. And it does look a little bit like a hairball. So the real question now, to me, well, one of the real questions is Arabidopsis a good model? Can we take this and move it into, apply it to other plants? And so that was the challenge, and particularly I didn't really want to take on that challenge, but of course when the Plant Genome Project got going at the NSF, Plant genome means any plant genome except for that of Arabidopsis. And so I needed to, if I wanted to get money, I needed to find a crop. And my friend and collaborator, Cynthia Weinig, said, got the system for you. I found a great population of recombinant A-bred lines in Brassica rapa. And so I said, okay, let's put in a grant to work on that. We did, and it got funded. And so now, many years later, we're still working on it. So transition to a crop, Brassica rapa, it's Arabidopsis on steroids. Arabidopsis flower, Brassica flower, Arabidopsis seed, Brassica seed, Arabidopsis seedling, Brassica seedling. So it's bigger, but it's very similar. And then there are a couple of other twists will come up. It's a cultivated crop, Napa cabbage, turnip, Chinese cabbage. It's highly pleomorphic. I'll show you a slide on that later. It's also an invasive weed, but it's closely related to Arabidopsis, but also to maybe a mega crop like being Canadian, of course, canola. So we thought this was a good place to start. And yeah, I didn't really realize at the time, but Brassica rapa doesn't transform terribly well, and that's really a limitation. But we wanted a nice, non-invasive, non-transgenic assay. And for that, we went back to the assay that drove circadian rhythm research for many millennia, that is leaf movement. And so there's a Brassica seedling, and this is what these guys do. Just the leaves go up and down once a day, for the cotyledons. The true leaves do as well. And I'll come back to this later, but we wanted to greatly enhance our throughput. When we first did our first recombinant inbred line population, it was 150 lines. We did it at 3 temperatures, and that took 3 postdoc years. And what we wanted to do was scale up and steal from Ed Buckler's playbook and start looking at nested association mapping populations and look at more. And so we wanted to do 1,050 genotypes, and I didn't have like 30 postdoc years. There was no way that was going to happen. So we had to streamline the analytic pipeline, and the problem is there was just too much operator input. And so I went to a friend of mine, this guy here, Hani Farid, who's chair of our computer science department. And I said, Hani, I know in your deep dark past you did automated motion detection. Do you have any code that, can you help us with this? And he said he thought he could. And so he wrote this great code that basically takes successive frames, a pixel, and draws a motion vector from where that pixel is in frame 1, frame n, to where it is in frame n+1. And that's what these blue lines are, these motion vectors going from frame to frame. And then you simply sum all the vectors at each interval, and then you can plot out a motion. And it's totally, user-free. You just click one button and you get the whole thing back from all your plants. So if we, oh, Ani, I want you back. So we plot that out and you get nice automated digital readout that's quantitative, and we can then do a Fourier transform and extract the rhythmic parameters on that. And so here are traces from the 2 parents of this recombinant inbred line population. This one is much taller, and as you can see, it's growing very quickly. But basically, they look pretty similar. They go up and down once a day. And this is what they actually look like. The oil seed is yellow, narrow, thinner seed coat, so you can pack more oil in there. The rapid cycler, IMB211, has dark seeds. And you can see this is bigger, but this guy's already flowering. It's a rapid cycler. So they're a little different. Recombinant inbred line population generated from these. And so first we wanted to know, is there variation among accessions of Brassica rapa in terms of circadian parameters? So we just went to a Dutch stock center and got 50 cultivated accessions, measured circadian period length at 3 different temperatures, and we saw quite a range of circadian period length. We then applied that to the 150 recombinant inbred lines, and in every case we see transgressive variation and a lot of variation. Every bit as much variation among these lines derived from the 2 parents that are indicated by the circle and the square, as we saw in this global collection. So we can map QTL. And so while we were mapping QTL, Cynthia was, for circadian periods, Cynthia was mapping QTL for all kinds of morphometric parameters and physiological parameters. And the idea was then we bring the 2 together and correlate them and ask, is clock function contributing to some of these other functions? When we did this, we observed a QTL at this position on chromosome 9. And so at this point, the brassica genome had just been released, and we had a sense of what candidate genes were there. And now I want to introduce one of the complications. Brassica rapa is just like a Arabidopsis on steroids, perhaps, but since the separation of Arabidopsis from brassica from Arabidopsis, it's undergone a whole genome triplication. And so we mapped out where all the various candidate genes were based on synteny with Arabidopsis. And we had a candidate underlying this one where there was only one copy had been retained. After polyploidization, you get a lot of fractionation, a lot of gene loss. We thought if we picked one where there's just a single copy, life would potentially be simpler. And the gene underlying that particular QTL was a gene called gigantea, which is known in Arabidopsis to be important both for flowering time, loss of function mutation is very late flowering, and also for circadian function. So we thought that's a good candidate. So the first thing we did was try and confirm genetically that there really was a gene in that region. And we were lucky in doing this. Normally you'd make nils or something like this, but we got the population of rills very early. They were only been selfed about 3 times. And so there was still a lot of residual heterozygosity. So we could take one of the rills that was heterozygous over the QTL and simply self it and generate 2 parents in an otherwise homozygous background with the parental DNA either from R500 or IMB211. In doing that, we had a recombination event that greatly reduced the interval. We could simply compare these 2 lines. And indeed, there was a statistically significant difference in period that was consistent. IMB211 has a longer period than R500. So, okay, there's something there. If gigantea were to be the gene that's responsible, presumably there'd be some sort of polymorphism between the 2 alleles. So we resequenced the 2 alleles. We found a ton of SNPs, 3 of which were actually predicted to be missense mutations, changing the amino acid sequence, a serine to alanine, and so on and so forth. So then we went right to the transgenic endgame. I told you we can't transform brassica terribly well, so we just went into Arabidopsis, put the brassica alleles into Arabidopsis. And we got very lucky and got a home run with the first experiment, because when we introduced the IMB allele into Arabidopsis, nothing happened to circadian period length. But when we introduced the R500 allele, and that's the one with shorter period in brassica, we had a significant shortening of circadian period length. There were 3 mutations, so we started making chimeric genes. And ultimately, the final thing, we simply made point mutation to recapitulate this mutation. And so the IMB211 allele that doesn't shorten circadian period length, if we simply make that one nucleotide change that amino acid to the R500 version, it now shortens period. And the converse, R500 shortens period, but if we change that back to the IMB residue at that position, fails to shorten period. So that's the gene, and that's the nucleotide, and that's the amino acid responsible for this. Biochemically, mechanistically, we don't understand. But that is the mutation. It's not like the 2 alleles, one is active, the other is inactive, because both alleles fully rescue the late flowering phenotype of a loss of function gigantea. So they're active, equally active for flowering timing. And gigantea turned out to be, we didn't know when we started this, but it's pretty interesting because it does a lot of things. We knew there was a hypocaudal phenotype difference, and everything that I told you about circadian period applies to the hypocaudal phenotype. Also, it turns out loss of gigantea function in Arabidopsis confers tolerance to cold, enhances tolerance to cold, and that's seen here. It's able to tolerate lower temperatures than wild type. And the IMB211 allele eliminates that, whereas the R500 allele does not, and the chimeric genes behave consistently. And there's a similar phenotype that I took out, apparently, with salt tolerance, and that behaves the same. So we wanted to ask, okay, that's all in Arabidopsis. Does this mutation matter and do the same things in Brassica rapa? And for that, I've always relied on the kindness of friends. And Lars Østergaard at the John Innes Institute had developed a spectacular tilling population in an oilseed RO18 that's closely related to R500. And so we sent him $2,500, and he sent us 22 alleles of Gigantea, including 4 of which were presumptive loss of function. And so here are 2 of them, and it's late flowering, a loss of Gigantea, late flowering, enhances freezing tolerance and enhances salt tolerance in Brassica rapa. So it's behaving quite similar to the Arabidopsis story. It also doesn't do much to circadian rhythmicity at 18 degrees, but at 22 degrees, really flattens out the rhythm. There's a big loss of amplitude. So it's clock active as well there. So we got a QTL. Brassica, as I mentioned, is highly pleomorphic. And if you look in Brassica rapa, it's all these various, there's turnips and there's leafy vegetables. Of course, we're more familiar in North America with the Oleracea equivalents, where it's cabbage and Brussels sprouts and cauliflower and broccoli and so on and so forth. And a lot of this may be because of this triploidization, providing the material for diversification. And of course, we know that gene duplication underlies the radiation of the angiosperms. Below all the important divisions, right before the ancestral gene plant, there was a whole genome duplication. Before the ancestral angiosperm, there's a whole genome duplication. If you're Arabidopsis centric, you're familiar with the gamma, beta, and alpha duplications on the road to Arabidopsis. But this little triangle up here in Brassica rapa is this recent whole genome triplication that's gone on there. And so typically these events are followed by a great deal of fractionation, a lot of gene loss. And so when we wanted to evaluate candidates for our clock QTLs, we wanted to map all the various Arabidopsis clock genes on the basis of syntenic position. And when we did that, we realized we could ask the question, are clock genes fractionated lost at the same rate as everything else, or are they somehow different? And Jim Birchler and Mike Freeling have elaborated this dosage hypothesis that says that genes that are highly networked or genes that are dosage dependent cannot be lost following a whole genome duplication, because you throw off that dosage balance. And of course, all the genes in the clock are semi-dominant and dosage sensitive. And so we thought, well, maybe they're preferentially retained. And when we started looking, we started thinking this might be the case. Here's one gene family of 5 pseudo-response regulators, PRR9, 7, 5, 3, and PRR1, which is also more commonly known as TOC1. And you'll notice for PRR5, there's 3 copies. For PRR9, there's 2 copies. 2 copies for PRR7. It looks like they're preferentially retained, perhaps. And so we compared the clock genes to the 10 syntenic genes around each of the clock genes. And the clock genes, about 60% of them are retained in rapa, whereas only about 40% of the syntenic genes were. So yeah, they are preferentially retained. And it could be that this whole genome duplication business in plants underlies the proliferation of clock genes. And it just empirically, when you look at the hairballs to describe the oscillator in animals versus plants, they look a lot more complicated in plants. And so if you look from a single-celled alga through moss to Arabidopsis to Brassica, you can just see a proliferation of potential clock genes. And so we're very curious whether or not this has, whether there's now neofunctionalization and subfunctionalization in rapa relative to what we know in Arabidopsis. I'm not going to talk about that today, but those are ongoing projects in the lab. So we wanted to describe the circadian transcriptome in rapa. And so we just set up to do a classic experiment. You entrain plants to a light-dark cycle, release them into continuous light and temperature. And we've harvested tissue every 2 hours for 48 hours using this oilseed R500. And starting to look at this. And so here's a set of clock genes. They're present, 3 of them are present in 2 copies in rapa. Third one's present in 3 copies. So just in this survey, and this is representative of what we see, and other people have seen, typically when you start looking at one of these polyploids, you can fractionate the genomes, and there will be a dominant genome and a subdominant genome, or genomes, where one of the genomes is expressed at higher level than the other. And we see that here, one copy of LHY, that MIB domain transcription factor, is expressed at higher level than the other. Same with TOC1, same with PRR7. The same is true with PRR5, where PRR5 is what we're calling the C copy here, is expressed at higher level. But now we start seeing evidence perhaps of some subfunctionalization going on, because you'll notice one of these genes, PRR5B, is peaking earlier than the other two, C and A. And so we don't know if these are now starting to subdivide functions up or acquire new functions, but certainly in terms of the pattern of transcript abundance, we're starting to see changes. And we see this in some, but not in all of the genes. So if you start trying to sort these out and the various clustering algorithms, we're using network analysis to look at this. One of the predominant things when you sort the transcriptome is time of day. And we can sort this out into clusters on the basis of time of day of the peak of transcript abundance. ZT, that's a Zeitgeber time. It's from the German time giver. Dawn, the last dark to light transition is ZT0, and then you scale from then. And if you're doing a light-dark cycle, you go ZT0 up to 24, which becomes 0 again. And so we can bin them out according to phase and see clusters of genes at all the different times. This is just the first few phases. We can start also doing network analysis, for example, in this family of the 3 PRR5 genes. And 5B that's peaking early is now in a network that's distinct from the 2 copies of 5C and 5A that are peaking a little later. And these have targets in gray that are in common, as well as own co-expressed, not targets, but co-expressed genes. And so we're starting to see some subdivision of the networks among the different copies. And we think that may be offering the opportunity for greater complexity in fractioning out genome expression, but we don't know yet. It's very early days. We wanted to start looking at response to biotic stresses. And I mentioned gating, the idea that the ability to mount a response will be a function of the time of day at which the stress is encountered. And so that's sort of illustrated here. If we provide a stress, say at this point, we might get one response. If we at dawn, we might get one response. If we provide it at dusk, we might get a different response on a basic transcript cycle. Generally, what people encounter when they do this is they see if there's a basal rhythm in the transcript, it tends to be that the peak in response to a perturbation tends to be at the time of peak expression. This was first observed by Andrew Miller working with Steve Kay, quite a while ago now, just looking at the light inducibility of CAB genes. And they peak in about the middle of the morning, and that turns out to be, if you put them in the dark and give them pulses of light, you trace their basal waveform on their inducibility, or their inducibility simply mimics that. When they're maximally expressed, that's when you get maximum induction. And so we're anticipating seeing something like this. And so we wanted to, and this has been seen by Mike Tomashow's lab, for example, in cold induction of the CBFs, where if you provide a cold pulse 4 hours after dawn, you get strong induction. If you provide the same cold pulse 16 hours after dawn, you get very minimal induction. And so there's this circadian gating of the induction of the cold regulon. So we wanted to look at drought. Why? Well, corn yields, if you look, whenever there's a dip, often that's associated with a dry year. And so we thought we could look at this. Also, Olivia Wilkins had suggested that there was a circadian gating of drought responses in both Arabidopsis and poplar. So we thought this would be a good place to look in Brassica rapa. And we know that there's likely to be a clock intersection from some Arabidopsis work. We know that if you perturb the expression of TOC1, either overexpress it or do a knockdown or a loss of function mutation, you see effects on stomatal aperture. In response to ABA, the closing is attenuated by TOC1 overexpression. We know that a triple loss of function mutation of 3 of the PRRs, PRR 9, 7, and 5, enhances drought tolerance in Arabidopsis. And so we thought that, there's good evidence suggesting there's going to be a clock intersection with the drought response. And so we went after this. Our collaborators, particularly Brent Ewers and Lena Guadagno at the University of Wyoming, spent a lot of time trying to get conditions where they could withhold water and get a gradual loss of soil water moisture and look in the earliest stages of the drought response. And that's what we're trying to do here. Here's soil moisture in well-watered versus our droughted plants over. We withheld water a day before this, so we're looking in the second and third days of drought here. And we're seeing only small changes in stomatal conductance in the well-watered versus the droughted plants. So it's a very early response. These are a long way from wilting. And so we're going to look, these guys, the Wyoming guys, are going to look at a lot of physiological parameters, and we're going to look at the transcriptomic response. And so they measured photosynthetic rate, stomatal conductance, photosystem 2 efficiency, non-structural carbohydrates. And there are not much for photosynthetic rate, but we see stomatal conductance changes with it depleted into droughted plants late in the day, sort of what you might expect. We see perhaps compensations in photosystem 2 efficiency, which seems to be slightly greater in the droughted plants, that maybe why photosynthetic carbon assimilation doesn't fall off. And we see some changes that they're significant, but I'm not sure I understand them at all in non-structural carbohydrates. But there are subtle physiological changes in the plants. When we look at the transcriptome, one of the things we see that's predicted from what Olivia Wilkins had done is that if you do a cladogram, the droughted and well-watered plants cluster together according to time of day. So there's a massive resculpting of the transcriptome in response to the clock. There's big time of day changes. About a third of the transcriptome cycles, depending on where and how you look. And so what we actually see then is the time of day signal is predominant over any response to this slight drought condition. And so what we're looking are for subtle changes. We're doing weighted gene correlation analysis, gene correlation expression network analysis. Yeah, and so we're going to split this up. We're going to make a droughted network, a droughted and a well-watered network. We're going to establish a consensus by comparing the two, and then we're going to look for modules, groups of co-expressed genes that respond and are correlated with the physiological data and unique to the droughted network. That's what we're trying to do, and it's very early days in this analysis. But we can set up, and the different modules are color coordinated, and we can do that with well-watered and droughted, and we can split them up into modules. The top 8 modules from each network contain about 85% of the 80 to 85% of the expressed genes in these top modules are cycling. That's the way to phrase it. And so if we can take a well-watered network and that shows, here's the eigen gene expression, it's peaking in the middle of the day. And then if we re-examine those genes, actually they've now subdivided into 3 subnetworks. One that looks much like the parental network, unperturbed, but 2, this network and this network. You can see a broadening of the peak in the daytime, and here's sort of a broadening and a phase advance of the trough. And so these are subtle responses in how, and of course we don't at this point know that they're functionally important, but we can start seeing perturbations in response to drought. So if we look at these physiological traits, we can correlate them with the expression profiles, and we can observe, for example, here's a group that's positively correlated with the physiology, with carbon assimilation. Here's an example of a group, a module, that's negatively correlated with Fe over Fm. In creating this curve, we flipped to an absolute take an absolute value, and that's why it looks like a positive correlation. It is a negative correlation. And then there's, of course, other modules that simply are not correlated with somatoconductance in this regard. What we're starting to see are things where, you know, they're peaking in the drought and not, or the amplitude of the peak is up in droughted versus well-watered conditions. And, you know, inspection by eye, when you look at this, okay, here's the pattern that I told you in the well-watered network. In the well-watered, this transcript is peaking, and the amplitude is increased under droughted conditions. But of course, inspection by eye is pretty unsatisfying. So we've tried a number of things. We've taken trying to identify rhythmic genes is one of the challenges in transcriptomic level analysis. And some people, John Hoganesh and his colleagues at Penn, have developed something called JTK cycle that is a way of evaluating the significance of rhythmicity and phase. And so we're comparing individual transcripts in the droughted and well-watered, and we're looking for transcripts that either are not rhythmic in droughted and acquire rhythmicity in well-watered, or are not rhythmic in well-watered and acquire rhythmicity in droughted, or the converse, or ones where there's an amplitude change, either up or down. And this program is working fairly well for pulling those out. So we're getting genes in that regard, and we can build networks among these genes. Here's, for example, a network of drought-responsive genes that are positively correlated with somatoconductance. And it's actually 3 overlapping networks. And there are, conversely, drought-responsive genes that are negatively correlated with somatoconductance. So we're trying to parse out the transcriptome in this sense. But our analysis is far from complete. And then, of course, the hard thing is going to be to relate specific changes with differential drought response. Now, when does this end? Okay. Okay, so we wanted to scale up, and I gave you my honey spiel about how we could scale up. And so we wanted to look at a lot of different plants across the eudicots. And so when we did that, we could observe in Arabidopsis and other brassicas, but we went into things like Emulous and as far as soybean. And it turns out that this new program that Hani wrote for us that we call TRIP, Tracking Rhythms in Plants, seems to be generally applicable across the eudicots. And that allowed us to investigate one of the things that I've been really interested in and started out talking with. Is there some sort of, can we evaluate a fitness effect of clock performance? And so we started out looking in soybeans. And you know, soybeans, it's a different type of rhythm, but the leaves go up and down quite nicely. And of course, you can get the motion vectors on it. And so we can do this. So what we did was we went to Monsanto and got 50 elite cultivars. And these cultivars have been extensively bred for basically flowering time and yield performance in latitudinal zones. They call them maturity zones, but basically they're grown in these latitudinal zones. And when we looked at circadian period in these 50 elite cultivars, that nobody's messed with their clock, nobody measured their clock, but we observed a significant positive correlation of period with latitude. As you move farther north, the period lengthens. So were the Monsanto breeders inadvertently selecting for circadian period length? Perhaps. Certainly all they were looking for was performance in these latitudinal zones. So we see this correlation. It was a great conceit to get it from Monsanto because, of course, no one's going to doubt that they're well-bred. The problem is it's the end of the road. They might know what the genes are, but we're not going to know what the genes are. The agreement didn't include access to that. But we've repeated this now in a wild population of monkeyflower, Emulous. And John Willis and Josh Suzy have collected accessions of Emulous that grows all the way from the Mexican border up well into Canada, the Queen Charlotte Islands. So a huge latitudinal range. And two what do you call it? Natural history lifestyle habits, annual and perennial. Among the annuals, there's an even stronger positive correlation of latitude and circadian period. So where we're hoping to go is to take these genotypes and start doing the common garden experiments and ask, does circadian performance contribute to fitness in the different latitudes? So that's where we want to go with this. We haven't done that yet. And we've expanded dramatically. The high throughput has allowed us to now look at large numbers of populations of Arabidopsis. And this is our all we're using now, it's, where are we? That's a Canon power shot. You know, wait till they're a year old, you go to Home Depot, they're cheap. You can get them, you hack in and overwrite the software and turn them into time-lapse machines. And what Hani's program allows us to do is capture 120 seedlings and then immediately convert it into individual seedling temporal image stacks, and then do the entire analysis and release period and phase data for that seedling and that seedling and that seedling with one click of the button. So the analytic pipeline is really smooth. And so we can fit 14 cameras into our growth room. And so here's an example of looking at 7 populations, all against a common female parent called 0. This figure is 4.5 million images, and it was about 30 times faster than the preceding one. So Hani is a genius as far as I'm concerned. And so you get what has been shown. If you analyze the individual populations, you get some QTL for circadian period, a fairly low LOD scores. If you do the combined analysis, the LOD scores go way up in significance because you've increased statistical power. And so now we're very interested in trying to identify some of these. This one we think might be our old friend Gigantea. This one, well, the candidate there is PRR7. This one in the middle of 4 is, as far as we know, novel. And so we're pretty excited if we could actually get a new clock gene from this quantitative approach. That'd be terrific. To do this with 1,050 genotypes, with all the replication, you know, we had to do randomized block design. And the postdocs, Katie and Ping, came to me and said, you know, we can't stand the idea of throwing away the seedlings after we've measured leaf movement. What else can we look at? And we thought about this, and we thought, well, let's go back to that example of starch. So we then took all these plants, grew them a little longer in a light-dark cycle, and then harvested them and sent them off to Mark Stitt in Germany for starch analysis and metabolite analysis. So now on the same population, we can measure circadian QTL and QTL for residual starch prior to dawn. And when we do that, they really dragged their feet doing it because they didn't think anything exciting was going to come out. And they discovered more variation in residual starch than, it was like an order of magnitude, more than they'd ever seen. And they were pretty excited. So now we have QTL for circadian period and starch, and sometimes it's one or the other, and sometimes it's both. So we're really hoping, Mark and Allison Smith and I have just submitted a proposal to pursue this, identify some of the genes, and really look at this interplay of the clock with starch metabolism. And it was amazing. In 6 of the populations, you'd predict if you had a long circadian period, you'd anticipate dawn farther out. So you'd be burning your starch at a slower rate, and you'd have more residual starch. And in every population, when you there's a positive correlation between period and residual starch. And in 3 of the populations, it's fairly significant. So it looks like that's borne out across these. And so we're pretty excited at pursuing this. So that's where we're going to be going. I never would have thought I would be interested in starch. But Mark and Allison have made have converted me. Back in Brassica, Rick Amasino and Scott Woody have been preparing a series of populations in this nested association mapping strategy. So we're hoping to repeat this type of work in Brassica Rapa. Of course, I didn't do any of this myself. It's the product of mostly some great postdocs, Katie, Victor, Ping, a former postdoc, Qi Guang. I mentioned Hani, a couple of visiting scientists, Daling and Jin from China and Korea, and a horde of really good undergraduates, current honors thesis student, and a whole bunch of collaborators. Our plant genome work has been with Rick and Scott and Cynthia and Brent and Lena and Todd and Malia, and then some collaborators in South Korea. And let's see, Ganesh and Cindy gave us the soybean cultivars, John and Josh, all the Emulous cultivars. And Mark, this is an optimistic collaboration. We're just getting started, and hopefully it'll lead to exciting things. And of course, I want to thank the National Science Foundation and the Rural Development Agency in South Korea for supporting this work. And I'd like to thank you for your attention. I'll be glad to try and answer any questions.
Questions?
Please.
Define circadian rhythms.
Yeah, that's a little controversial. Hugh Nimh did an experiment that suggested mutations in CCA1 and TOC1 didn't affect the root clock. People have revisited that, and there is a root clock. And it looks like there is a lot of, a guy named Endo, I forget his first name, Japanese fellow starting, it was a postdoc with Steve K, he's got his own lab in Japan, has been looking systematically. And there are different, the clocks in different tissues are functioning slightly differently. So there's an epidermal clock, and a mesophyll clock, and a vascular clock, and a root clock. And then Paloma Mas did in Barcelona did a really interesting experiment where she excised the shoot apical meristem and discovered that the root clock stopped running. And so it looks like there may be some hierarchical relationships among these. So that's a very active area. But yeah, there is a root clock. We knew that I didn't show you these data, but I've been collaborating with my wife, Mary Lou Garino, who's Melina's buddy, and she studies iron homeostasis. And it turns out I would always be a pain in the butt at every one of our student seminars and say, what time of day did you do the experiment? And they would say, shut up, Rob. And so I would. Finally, my Korean postdoc got together with her Korean postdoc and finally did the experiment. And the reductase that reduces the iron and the transporter that transports the iron in both show they're strongly iron inducible. So you only see this if you're doing under iron starvation conditions, but they're circadian gated. So they peak dramatically at one particular time of day. So yes, the clock is regulating iron homeostasis in the root. So there clearly has to be a root clock. And so we're collaborating on trying to figure out how that works, but we're still pretty much in the dark.
Just to add to it, we actually had the same experiments that in roots, we were trying to analyze gene expression in response to copper deficiency, and we found like the data were all over the place. And then we fixed the time of RNA collection, and all of this variation just gone. So it's that.
It turns out this experiment has been done over and over. Many of my collaborators and colleagues in the circadian field started out doing gene expression, and one postdoc couldn't reproduce the graduate students, and one was a lark and one was an owl, and they fought like cats and dogs, and then suddenly they realized they're both right. And so that has dragged a lot of people into the clock field, and kicking and screaming typically.
Yeah, I think it's a pretty related question. I'm just wondering, so which organs or tissue are you collected for detection about the related genes, and whether there is any difference among them to censor the environment?
Yeah, if you start doing, laser dissection or protoplasting and sorting, you can start seeing in these epidermis, mesophyll, vascular tissue. In the leaf, for example, you can see slightly different profiles of gene expression, and different mutations affect different clocks, different tissue-specific clocks. So we haven't done any of that ourselves, but that's definitely a growth industry in the area. And everything I've shown you and everything I've done is a leaf lamina. So predominantly mesophyll, but with epidermis in there. And we showed, Todd Michael, when he was a grad student in my lab, did some experiments where he provided light-dark cycles or temperature cycles, and he would put them in opposition to one another, sort of this gladiator experiment, and ask which is more important. And it turns out the answer depends on which target you're looking at. So a mesophyll expressed gene like CAB responded primarily to light rather than temperature, but an epidermal catalase responded primarily to temperature rather than light. And so there's going to be a great deal of complexity there that is only beginning to be explored.
Yeah, so there are so many genes involved in keeping this oscillating network going, but when you look at QTL, you get, a few big peaks, it looks like, and some of them are genes that you know are involved in this, like Gigantea. Is there anything about those QTL genes and their position in the co-expression network? Are they more connected? Do they occupy like transitional positions between modules that's informative, or?
We haven't seen that yet, but basically, you know, it's big intervals and very few of the actual genes have been identified. Seth Davis has identified ELF3 in one population, and we've identified Gigantea in a different species in a population. We have a good candidate for one in Arabidopsis now, but so we don't know enough to know about that. But also, you know, the log scores are misleading because each one of those, you know, I wish we had, you know, a really large effect gene, but each one of these QTL, I think the largest one we've seen is accounting for 10 or 15% of the variation in the trait. So there aren't, these are definitely not behaving like Mendelian loci. You know, they're not large effect. So makes it more challenging, but perhaps more interesting. And you've exactly articulated part of our interest in pursuing this. Will that turn out to be true? Love it if it were. Susan.
So you said you were showing that decline of the Monsanto soybeans. So do humans show a decline in clocks, or does the Icelandic population, by and large, have a different kind of clock, or have a different? I mean, I know you say we can train, but I'm asking whether there is a genetic component to the clocking.
I don't know of any study that's been looking geographically in that regard, but we do know that there's a genetic basis in the clock for a number of human pathologies. So advanced familial sleep syndrome and delayed familial sleep syndrome. Underlying that are mutations in canonical clock genes like PERS, BMALs, and so on. So whether or not there will be a clock function thing, I don't know if we've been in those latitudes long enough, but it's a really interesting question to explore. And people are starting to do this more and more. They've had to really, it used to be when I started in the field, they thought that we all ran really quite long. And that was, you know, the way they did the studies, as it turns out. It was an artifact because they take somebody, typically a PhD student who wanted to write their thesis, or a professor who wanted to write their monograph. And so they wanted to be left alone. So they'd volunteer for one of these cave or bunker studies. And they'd go, you know, underground for some period of time and write their thesis. And then when they'd open the door to let them out, they'd say, hey, no, come on, I'm counting the days. I've got another week to go here, and I haven't finished my last chapter. But that's because the clock was running a little long. But they gave them control of the light switch. And it turns out that lights in the evening delay your clock. So just with the presence of light in the evening, they were actually artificially lengthening the period of their clock, which is, of course, why you don't want to do your email right before bed. And so now to do these studies, it's not so fun because they strap you down in a dim room and intubate you and, you know, put an IV in and give you 200 calories every, you know, et cetera, et cetera. And our period is much more, as a species, is much closer to 24 hours than we thought. And I don't know what the range is. I don't think they've done it on a lot of people yet. But it would be really interesting to look at. I mean, maybe that's why the Scandinavians are depressive or something. I don't know. Maybe it's because there's no sun, or manic depressive. Right now, I'm kind of moving into the manic phase. I love this time of year. And maybe that's just because it's been so dark for so long. I would think the Hanover weather is much like the Ithaca weather in that regard.
All right. Well, before thanking Rob for the seminar, there's an announcement for the student. There are openings for lunch, so please come see us after the seminar. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.